To begin our session, we will be giving the reins to Dr. Nikki Heherson de Gamac, our moderator for this afternoon. Sir Nikki, please go ahead.
Yes, thank you, Sir JC, for the introduction. So I again would like to welcome everybody and all of our participants for today's afternoon session of our fourth UST-USP Symposium. So for our first part of this afternoon session, we will have two speakers that will tackle on the topic Science in Education in the New Normal. So let me first introduce our first speaker this afternoon session. Our first speaker is an experienced faculty member with a demonstrated history of working both in the basic education and in higher education industries. He is skilled in program evaluation, instructional design, curriculum development, and curriculum leadership. He actually finished his Doctor of Philosophy, focused in curriculum studies at the University of the Philippines, Diliman. He is currently affiliated as a faculty member of the University of Santo Tomas Senior High School, and he currently also served as an editorial review board member for Site Journal of the Association for the Advancement of Computing in Education and the Journal of Science and Mathematics Education in Southeast Asia of CIMEO RECSAM. His research interest areas include curriculum implementation and teacher agency, curriculum studies, pedagogy and instruction, STEM education, and science education. Ladies and gentlemen, to present his paper entitled, Are They Really Learning? Harnessing the Power of Formative Assessment in Science Classroom During COVID-19, may I present to you Dr. Louis Dasas.
Good afternoon. I am Louis from the University of Santo Tomas, Manila, Philippines. I'm here to present the preliminary parts of my paper entitled, Are They Really Learning? Harnessing the Power of Formative Assessments in Science Classrooms During COVID-19. First, I'll talk about the background and rationale of my paper. Then, I'll give the preliminary findings and my insights based on these findings. The COVID-19 pandemic challenged existing educational systems, leading to curriculum delivery disruptions. This forced schools to shut down and teachers rapidly transition to using emergency remote teaching and learning in order to deliver the curriculum. The pandemic teaches us various lessons. Morelli argues the centrality of teachers and the immateriality of physical schools at the time of pandemic. Anove posits that we must not return to the banking model of education and curriculum that is clouded by knowledge deposition and heavy testing. Instead, UNESCO suggests the need to approach curriculum and all related matters, such as assessment, differently. As we continue to unravel the challenges of remote teaching and learning, I ask a thought-provoking question. Are the students really learning? Teachers must understand how classroom assessment can be best implemented in a distance education setting, since teachers are at the forefront of implementing distance education. Classroom assessment in distance learning must not solely depend on summative assessment practices one used to do in an in-person class setting. Teachers must remember that teaching remotely follows an entirely different paradigm. Allow me now to present to you this table that differentiates traditional face-to-face learning and remote teaching and learning. As you can see, in traditional face-to-face learning, the role of the teacher is to manage information and be the source of content. However, in remote teaching and learning, the teacher shifts role to becoming a curator of relevant content and designer of learning experiences. The teacher must be knowledgeable now on what content to present and what learning activities and experiences to provide for the students in order for them to learn the concepts. In traditional face-to-face learning, there's structured learning environment. There are fixed schedules, classrooms, tables, and chairs. However, in a remote teaching and learning setting, learning is more flexible. It can happen anywhere, anytime. In traditional face-to-face learning, there's limited interaction provided for by the one-size-fits-all delivery, usually a lecture. However, in remote teaching and learning, there are more opportunities for interaction given the varying modes of delivery. For example, a teacher can deliver a lecture via podcast, video lecture, or a live web conferencing application such as this one. With these in mind, teachers must also realize that classroom assessment requires a paradigm shift. And a paradigm shift requires an understanding of the three paradigms of assessment put forward by Serafini in 2000. These include assessment as measurement, assessment as procedure, and assessment as inquiry. Assessment as measurement is grounded on a positivist view of learning that regards tests as central objective measure of the amount of knowledge the students have acquired over time. This paradigm views knowledge to exist separately from the learner, and the learner needs to acquire knowledge and not necessarily construct it. The second paradigm, assessment as procedure, focuses on methods and strategies and not the epistemology of assessment. Like the first paradigm, it makes use of tests as a basis of learned knowledge with an addendum of some qualitative measures such as portfolio. The first two paradigms see students as external to decision-making processes in assessment. The last paradigm, assessment as inquiry, on the other hand, is based on a constructivist point of view, wherein students are involved in the assessment procedure, providing a deeper understanding of their learning and learning contexts. In short, assessment is situated. Assessment results are used to direct curricular decisions and facilitate learning. Formative assessment can be viewed as assessment for and assessment as learning. With reference to the previous paradigms I just mentioned, these are assessment as procedure and assessment as inquiry. Assessment for learning is a type of formative assessment that is utilized by teachers in order to gain understanding about students' knowledge and skills, and these information are used to guide instruction. On the other hand, assessment as learning is also formative assessment, but focuses now on teaching the students metacognitive processes to evaluate their own learning and make necessary adjustments. Remote teaching and learning rely heavily on teachers' use of formative assessments. However, these formative assessments do not necessarily function as informing instruction. Formative assessments should function to provide specific and immediate feedback to students on how they are learning. Recent literature in science education revealed that enactment of formative assessments remains diluted and undernourished. Johnson et al. Report teachers' inconsistent use of formative assessments in the classroom. In addition, teachers rarely use technology for the purpose of formative assessment. Similar findings were reported by Blacho in 2019. It was found that science classroom assessments were largely teacher-led and directed, with very minimal involvement of students. Science classrooms remain focused on using summative assessments instead of formative assessments. Espiritu et al. In 2018 argue that teachers use formative assessments partially instead of extensively. This can be explained by the fact that teachers' understanding of formative assessment is parallel with summative assessment. That is, both of them function to determine what the students have learned and both must be graded. Recognizing that there is a need to examine science teachers' formative assessment practices and how they influence student achievement during COVID 19, this paper aims to 1. Document a science teacher's formative assessment practice during COVID 19, and second, examine the effect of formative assessment on student achievement. Data about the science teacher's formative assessment practice was obtained using self-learning modules, which was analyzed using Dini et al.'s coding scheme. Additionally, formative and summative test scores were obtained and analyzed quantitatively. Formative assessment enactment of teachers was described in the model put forward by Sevian and Dini. The formative assessment enactment model serves as a guide in examining formative assessment practices of teachers, and it also provides researchers with a tool to study formative assessment enactment. It is composed of three enactment paradigms, namely teacher eliciting, teacher noticing and interpreting, and teacher advancing. This paper conducted document analysis of self-learning modules designed and implemented by a science teacher during the emergency shift to remote teaching and learning. The following formative assessment enactment paradigms were used to analyze the modules. The following are examples of formative assessments from the self-paced module analyzed. As you can see, there are variations such as making connections, making a mental model, listing their own understanding, listing essential concepts and processes, and also reflection questions. Analysis of the self-learning modules revealed that enactment of formative assessments adhered to the teacher eliciting and teacher noticing and interpreting paradigms, TE and TNI. Most formative assessments focus on what students know and how concepts learned from the modules can be understood and were understood by the students. Formative assessments also assess students' ability to draw relationships between and among these concepts. It was found that there is a variety of formative assessment strategies, which included um, word association, graphic organizers, mental modeling, journaling, idea mapping, and self and peer assessment. Formative assessments also included traditional paper and pen tests. It is interesting to note that the design of the formative assessments was scaffolded, providing ample prompts and support for students. For example, after reading and viewing a material on a biochemical process, students were made to construct a mental model of this biochemical process. Before doing so, they were first made to list down key concepts and describe these key concepts. This allowed students to focus on key concepts and their relationships before constructing a mental model of the biochemical process. It was also found that teacher advancing, or TA, required structured and protected time. This was not observed in the self-learning module analyzed, primarily because the self-learning modules were self-directed. Sparling describes self-directed learning module as a comprehensive learning experiences that is well defined, inclusive of tests and resources that is usually done by the learner independently. Additionally, teacher advancing or TA enactment was difficult to do because of limitations of technology in terms of the teacher and the student. The sudden shift to emergency remote teaching and learning required flexibility in formative assessment practices because of the challenges of lockdown and pandemic restrictions. Therefore, it is important to realize that teacher advancing enactment paradigm requires structured and protected time, where teachers can provide personalized and focused remediation strategies to the students. Formative and summative Test scores were quantitatively analyzed to examine the effect of formative assessment on academic achievement, represented by summative assessment score at the end of the quarter. It was found that on average, students scored 76.75% in formative assessment, while they scored 90.30 in the summative assessment. Interestingly, it was found that 88% of the students had better summative test scores compared to their formative assessment test scores. A simple linear regression was calculated to assess the ability of formative assessment scores to predict the summative assessment scores. Preliminary analyses were performed to ensure that there was no violation of the assumption of normality and linearity. A significant regression was found with an R squared of 0.278, and students' assessment score was found to increase by 0.256 for each formative assessment. Overall, it can be said that the improvement of student achievement or student summative assessment score was mainly attributed or can be associated with the use of formative assessments. Considering the findings presented in this paper, how do we view now formative assessment and student learning? Is there promise to using formative assessment? Allow me to use a plantito reference of growing plants. Plants need quality nurturing in order to robustly grow and flourish. In the same manner, the quality of formative assessments allows students to learn and achieve better in the classroom, especially at the time of a pandemic. Digressing from an everything is graded view, science assessment in the new normal must consider a processual paradigm, process oriented, with formative assessment at its core. Teachers must recognize the value of formative assessment in distance learning. Formal and informal assessments can be valuable tools in supporting student learning. Formal Formative assessments involves collecting evidence about learners' capacity, taking time to interpret this data, and incorporating it in an action plan. This can be in the form of curriculum embedded or planned for classroom instruction formative assessments. On the other hand, informal formative assessments, which I would like to call on-the-fly assessments, requires on-the-spot modification of instruction based on what is necessary and what the learners would demand at that particular moment. Now, despite some evidence of the power of formative assessments, I would like to put forward Dunn and Mulvennon's argument that the effect of formative assessment, especially in the new normal, must be carefully studied from a situational stance. Therefore, there is still an arising need to develop more coherent frameworks in harnessing the power of formative assessments in the classroom. It is also significant to explore how formative assessment impact those who need them the most, the low-performing students. After all, formative assessment in science education must not be viewed as predictive, but rather reflexive and reflective. The famous educational measurement expert Rick Stiggins stated that if we are finally to connect assessment to school improvement in meaningful ways, we must come to see assessment through new eyes. Allow me to end my very short presentation this afternoon with an excerpt from the seminal work of Black and William. Formative assessments allow teachers to determine where the learner is going, where the learner is right now, and how to get there. After all, it's not just getting there, it's the process of getting there. That concludes my presentation. This is the Here is a list of my references. Thank you very much for listening.
Thank you very much, Dr. Dasas, for that very informative talk and sharing the power, no, of utilizing formative assessments, especially on our science classrooms. So at this juncture, let us now proceed with our next esteemed invited presenter this afternoon. So we are very lucky that our next presenter or invited lecturer comes from not in the Philippines nor in Japan, but actually he is a part of the Department of Plant Protection in Bogor Agricultural Institute in Indonesia. He finishes his master's degree, major in biology, also at the same institute, and obtained his doctoral studies in entomology in Clemson University in the United States of America. Our esteemed speaker was able to work on research areas in genetics, particularly, for example, on genetic diversity and population structures of certain insects, wherein they were able to infer mitochondrial DNA and microsatellite markers and host range and population densities of giant white flies on horticultural crops in West Java, Indonesia. So, ladies and gentlemen, to present his very interesting work entitled Maintaining Connection, Schooling in New Normal, let us give a warm virtual applause to Dr. Yai Munara Kusuma.
Hello everyone. We are pleased to be joining all of you in this symposium. First of all, I would like to thank the organizers of this symposium for the honor given to us to share our experiences in conducting lectures and research in the new normal era. Today, I would like to talk about COVID-19 pandemic situation in Indonesia, policies from the Indonesian Ministry of Education and Culture, policies from IPB University, teaching and learning activities at the Plant Protection Department in IPB University, the student final project, and we also want to share various example of practicum assignment. COVID-19 was detected in Indonesia in March 2020. Initially, its development was a bit sloping, but entering August 2020, cases of COVID-19 increased sharply and remained high until February 2021. The Ministry of Education and Culture of the Republic of Indonesia issued a policy regarding learning activities during the COVID-19 pandemic on some principles. First, the health and safety of students, lecturers, and education personnel must be a major concern. Second, the campus should not become a new cluster for the spread of COVID-19. Third, the academic year continues according to the N16 academic calendar. And fourth, learning continues by online. And this is new for us. And this is a good opportunity to share our experience about things that going online right now. The policy from the IPB University, first, the teaching and learning activities for undergraduate and postgraduate programs are carried out via online through LMS, online meeting platforms, etc. The final project of undergraduate student is carried out by conducting research at each student's location, secondary data research, or literature review, while postgraduate student final project or thesis that requires laboratory work can be carried out by implementing strict health protocols. Teaching and learning activities that require direct practice and cannot be fully implemented effectively through online learning will be carried out offline or face-to-face when time allows through integrated practical activities, thematic training, or other activities. Teaching and learning activities at the the Plant Protection Department. The number of subject courses held each semester. In an academic year in IPB, learning activities are divided into 2 semesters. The odd semester usually started at the end of August till the mid of January, and the even semester usually started early February until the 3rd week of June. The number of courses offered per semester for undergraduate program at the odd semester, there are 17 courses, and the even semester about 15 courses. And for postgraduate program, in the odd semester there are 31 courses, and in the even semester there are 12 courses. So the number of courses are still the same as before the COVID pandemic. Generally, the learning process in courses is carried out in 2 ways. First, lecture, which is delivered in one direction from lecture to student, and second, practicum, which is delivered using student standard learning methods, such as laboratory work, greenhouse, and field work. For example, for the course of Principles of Plant-Based Science, this course has 3 credits consisting of 2 credits of lecture and 1 credit of practicum. 1 credit of practicum is equivalent with 3 hours work in the laboratory, greenhouse, or in the field. 80% of odd semester subject courses and 85% of even semester subject courses are offered with practical content or practicum. I would like to show the comparison between normal versus new normal delivery methods. For lecture, during the before the pandemic, courses delivered in was by face-to-face between lecturers and students in the classroom. And during the pandemic, face-to-face activities between lecturers and students is in the virtual classroom using several online meeting platforms. And for practical work in usually is carried out in the laboratory, greenhouse, or crop field using real specimen of insect, pathogen, and plant infested symptoms. Whereas during the pandemic, practicum has to be modified. One method that the practicum were carried out at home using images of insect specimen, pathogens, and plant infested symptoms, or observation of pests or diseases around the house or in the area or where the student lives. Practicum also carried out by literature study from the internet or by communication between lecturers and students using social media. For example, we use a WhatsApp application. Now I'd like to talk about student final project. There's a comparison between normal and new normal. So the final project for undergraduate during the normal era or before the pandemic, laboratory was done. Research was carried out in the laboratory research, greenhouses, or field research in campus area. Whereas during the pandemic, the field research at a location near the student home, research using secondary data or literature review. However, for the graduate student, they have to do their research. So the research is, even during the pandemic, they do their research in the lab or in the field, but usually, if they do it in the pool, they usually do it not too far from their where they live. Now I would like to share some example of practicum assignment. Here we can see that the student work on in this example, in this picture, the student sent a picture of praying mantis, and they ask question whether they need to rear the insect to observe the biology of the insect. And they decided to rear the insect, and 5 days later, the student informed that the insect has laid eggs, and the lecturer instruct the student to put each egg or young mantis singly in a cup to avoid cannibalism. This is another example of a conversation between lecturers and student. The student informed the lecturer that they've found some symptom, and this is how they discuss what they found, and the lecturer or the professor gave instruction what to do with the specimen the student found. And this is another example, WhatsApp chat between student and lecturer, discussing about assignment of a particular work. So this is what we use because it's simple, and I think most of the student or every student has access to this WhatsApp application. Another way to do our practicum during pandemic is, giving them digital image of insect specimen or sketches. So the student assignment was to draw and to label the specimen they're drawing. The student redrawn the digital image that was sent by the professor, and they put a label on each drawing and explain what they found or what they see or what they draw during the practicum. Another method that we use for the practicum is survey of plant or pests or diseases around the students' home or in their area. They take picture of plant that show symptoms. For example, this is the, I think, infected corn, beans, and peanuts. And another way to do practicum is they make a very small project. For example, this one is baiting fungi, soil fungi using cucumber to by putting the cucumber on a damp soil, and they recording the growth of the fungus from that is in the soil. This is another example. the procedure of making media, PDA, for fungi. They use boiled potato with sugar and agar. This is very simple way to make growing media for fungi or bacteria. Here's another example of practicum assignment. The introduction of plant mycology. So the student saw seeds on a cup, and they have to observe for the growth of microorganisms. Another example is isolating seed-borne fungi, finding rhizomorph, and designing poster for international meeting. For integrated pest and disease management course, usually students have to do a comparative trial to determine the best practice for IPM. The pandemic situation made this trial impossible to do. So as a substitute, student group were constructing an IPM strategies by exploring many information from literature about pest and disease in some commodities, natural enemies found in the field, practice existing controlling method, and they discuss alternative controlling strategies after analyzing the bioecological aspect of the ecosystem. And they make a poster or the presentation about that. This is how we record our attendance. We use IPB mobile for lecture, for students, even for parents. From this application, we can see the number of students attend on lecture or practicum. So this is all we want to share. I hope that you can learn from each other, especially we would like to learn from your experience during this pandemic era. Thank you.
Thank you very much, Dr. Kusuma. May I again invite our two speakers to turn on their cameras, and I also would like to acknowledge the co-author of Dr. Kusuma on his paper that was submitted, Dr. Ali Nurmansha. Pleasant afternoon to you, sir. And also to our Japanese professors who just joined us into today's online afternoon session. So good afternoon, everybody, especially to our colleagues at the Department of Biological Sciences in UST. So at this juncture, probably we are now opening the online platform or the online floor for everyone or who likes to share their experience, particularly on this topics about the science education in the new normal, who likes to make a commentary or even ask questions to our esteemed invited speakers. So you can either type it at our chat box or even press the raise hand button here at the Zoom to be acknowledged. Okay, maybe I will throw the first question since I'm very interested on this theme that our two guest speaker provided for this afternoon. So it's one thing actually to do lectures in face-to-face. I think it's much easier actually. But now when we shifted to the online platform, obviously It's a common experience of most of us here, even in Japan, in the Philippines, in Vietnam, Indonesia, that it's quite challenging. Especially one thing that I find very challenging is giving feedbacks to our students. So especially during when we have this too much time, no, to give lectures, and then we wait for them for their submissions. So is there anything that you can share or tips or considerations um, on teachers like me, no, on how to do really effective and efficient feedbacks to our students?
I have a question.
Okay.
Okay.
Yes, Sensei.
Uh, yes. Uh, Thank you for your interesting presentation. I have same question to both two speakers.
Mhm.
How many students are instructed by one teacher?
Mm.
And in my case, it is problem. If the student number of students is too much, it is very difficult to answer each questions in detail.
So probably we can ask first. Maybe let's answer the first question I throw, no, about feedbacks. What is your opinion there, Dr. Louis, about effective and efficient tips of giving feedbacks?
Thank you, Dr. Miki. I think we're all guilty of that, no? The timeliness of feedback. So, two characteristics of feedback should be it's targeted, meaning it specifies how the student can improve, and number two, it's timely. So, it feedback should be given before, ideally, another graded or summative assessment will be administered. But this actually goes in contrary to the principle of flexibility in distance learning, such that we are flexible to accommodating students' outputs even beyond the deadline because of the restrictions that we have. Perhaps how we can possibly provide feedback is to give small but timely feedbacks for the students. I find oral feedback a bit helpful rather than the lengthy ones. And oral feedback, for example, I tried in my capstone class, I give the feedback by, instead of writing, because I also get tired of writing the feedback in Word, I just voice record myself. Okay, pointing out specific matters about their paper, which I can readily send to the group. In terms of Ma'am Emiko's question, that's one of the challenges that we have here in the Philippines, the large class size. I'm teaching to a group of 45 students. Yeah, that's too much now. That's really a lot. So one of the strategies that I devise is I divide the class session into two. So I half the class. So I meet the first 20, 22, and then I meet the other 22. It's quite tiring, but feedback is more effective and interaction is more provided because I get to ask everyone, I get to listen to everyone. In I call it the shadowing of a big group. When you're part of a big group, students tend to shadow out and try to be disengaged more. When you are a smaller group, there's no choice but to participate and to actively listen to the lectures and discussion. Thank you, Doc Nikki.
Hi, Nikki.
Yes, sir, sir, sir.
Just so to answer as well. Sorry. Okay, sorry.
Okay.
There you go. To give some insight as well to what Harada Sensei said, uh, Dr. Louis noted that in the senior high school, it's around 40 to 45 students. In college, particularly in the College of Science, lecture classes usually span the same way, around 30 to 40 students. But in the laboratory, one good practice is that when students go beyond 30, there should be a laboratory partner. So that's a bit helpful for us. In the graduate school, it becomes a bit more manageable. Um, Usually there's just around 5 to 10 students in the classroom. I'm interested, I'm curious. How about in Indonesia, in Bogor University, Dr. Yai and Dr. Ali?
Okay, should I answer first or Pak Ali? Okay, well, I can answer for two questions from Dr. Dagamak and Sensei Harada. In IPB, in normally in one class, actually there's 100 students, so around 100 students, 90 to 100. But we divided the class into two parallel class. So in one lecture is about 40 to 50 students per meeting. So there are two lectures, two meetings with different lecturers with 40 to 50 students per class. However, in the lab work, we only have 20, usually 20 students per practicum, so that's not too many. And in terms of feedback, actually it's kind of, we have a kind of problem because, you know, since not every student have a very good internet connection, so we have to, they don't have to turn on their camera. So they mute the microphone and they turn off the camera. And we don't know whether they're still there or go somewhere else or maybe fall asleep. But, you know, I usually give them a very short quiz just to check whether they're still on or they left. So, and it's sometimes a written quiz or just oral quiz. So we can see and measure ourselves if we can really deliver the topic and can be understood by the student. I think that what happened, but you know, if you see the title of our presentation is the maintaining connection. Actually, there are two meanings there. Maintaining connection in terms of that we have still school, we still have school. I mean, we still study. And also the most important thing is that, do they have any connection with us? Like, do they have good internet connection? Because almost in every class that we have, there's a lot of students just come in and join and left because they have a very bad internet connection. So that's the problem that we have here in Indonesia, because you know our students is from all over the country in the urban area and in rural area. Then sometimes they live in remote places and they don't have a very good internet connection. They just use their cell phone for attending classroom. So that's the problem. That's why we have the title maintaining connection. So we want to maintain our relationship with students academically, and also we want to make sure that they have a good internet connection. Every student in Indonesia, they get a free internet quota from the government, about total 50 gigabyte per month. It's free from the government to help the student maintaining their connection with the school. But again, the problem is not the quota. The problem is the connection. So I don't want that we student have to climb a coconut tree just to find a signal, for example. I think that's what happened. That's our experience here in Bogor. So thank you. Maybe Pak Ali, and we have Ibu Hagia here also, the co-writer of the paper. Maybe Pak Ali and Bu Hagia want to add something. Thank you.
Okay, Pak Yai, I just want to add what the Dr. Yai mentioned about the feedback from the student. Despite of the Pak Yai mentions, usually our lecturer to know how much student can accept the material of the lecture, in every week meeting, lecturer give assignment for student, and then 1 week later the assignment should be submit to the lecturer, to the to the lecturer, and then we know how much the student can get the material from the lecturer or practicum, and also every mid semester our university have evaluation system for the teaching and teaching and learning process. But in the especially in the pandemic like now, despite the evaluation system, IPB University also conduct a survey for all student, what the method of the online delivering for practicum and student, that student can have all this difficulty. And so from that, the information from the student and university shared to the lecturer, so we know what the problem in the student for the pandemic area. Yeah. That is, thank you.
Thank you, Pa Ali. Okay. Are there any more other commentaries or questions that would like to be raised? So far, what I'm getting in this discussion is that it seems that the number of students in our class is very important, all right? For us to give a very effective feedbacks to our students. Maybe I can also share some of what we did in one of our laboratories in evolutionary biology. So like what Sir JC have mentioned, like we have like if the number of students in our class in the laboratory reach 30, there will be 2 instructors in the teaching the course. So what we did is that we split the class for each instructor handling the group. So that it's like now one teacher will handle 15 and the other one will handle the other 15, and they do small group discussions wherein they will now at the same time they ask questions, and they also give live feedbacks to their students during synchronous sessions of our laboratory. And during some we also get some feedbacks from our students, and they said that it's much effective because now they feel that they are connected to their laboratory instructors and not just giving simulations or tasks or even just sending them message on the messenger. They see them and they feel connected because it's a small group, and then they are actively encouraged to participate. So that's one technique that we have been doing, that we are doing in our classroom. Okay. And another thing, it seems that in our chat box, it seems that connection, not just with the instructor, but as well with the internet connections in our countries, is one challenge, right, that we are facing. Yeah, okay. I don't know why, but maybe do you have any ways how you have able to handle or cope up with that situation, especially when students starts to complain that they cannot attend the class because of the bad internet connection? How did you handle those situations?
Well, in our case, we do not really ask the student to attend at the same time. Sometimes, if they do not have a very good internet connections, I can tell them to download our material from the LMS, Learning Management System online, so they can download the material over there. And actually, even though the classroom is only 2 hours, we give the meetings usually last at least 1 hour, 40 minutes to 60 minutes, because if it is too long, it's just so tiring just looking at the screen. And the asynchronous class is still very long. It's make us it takes more time. We discuss through WhatsApp, and we have group for every course, and you know, discussion can last for days, and you know, we need to respond to the question they ask, or to discuss things in the group. So it is a bit tiring. It's our, I think for most of us, it's the first experience, and we don't have really experience how to give, especially for practicum. Especially for practicum, it's like for us in agricultural university, it's hard to convert from field work to keyboard work. It's very hard. Okay, thank you.
Yeah. Thank you very much, no, Dr. Yai, for those insights. But I think what we're doing now, we're coping up with that challenges in our education system. So
We're learning.
We're learning. We're a work in progress, I would say. So thank you very much, Dr. Kusuma and Dr. Dasas. And that wraps up our session about science education in the new normal. Let us give again a very warm round of a virtual applause to our two esteemed speakers. So thank you very much, sirs. And now, at this juncture, we are now moving forward to a new theme for our symposium. So likewise, we will hear two talks on the topic about aquatic ecology research and education. So let me now introduce our third invited speaker. And she is actually a dear and esteemed professor from the University of Shiga Prefecture School of Environmental Sciences with expertise on freshwater biology, ecology, benthos, and parasitology. She graduated in the prestigious Kyoto University Graduate School and is an active member of various academic societies such as the Japanese Society of Parasitology and Ecological Society of Japan. Among of her vast research team that currently works includes the influence of population genetic structures on the trematode susceptibility of snails endemic in Lake Biwa, ecological control of introduced bucephalid parasites pathogenic to freshwater fishes, and the taxonomy and life cycle of freshwater digeneans. Ladies and gentlemen, without further ado, let me introduce our speaker who will give a talk about the example of remote field lecture of environmental science for first-year students of the University of Shiga Prefecture, Dr. Professor Misako Urabe.
Good morning, everyone. I'm Misako Urabe, the School of Environmental Sciences, the University of Shiga Prefecture. Today, under the subject of this session, Challenges in Science Education and Research in the New Normal, I will introduce our trial of a field lecture for first-year students in 2020 academic year. In this presentation, I'd like to emphasize that lockdowns are not only difficulties for the normal education program, but can be regarded as a unique chance to observe ecosystem change with less human impact than usual condition. Therefore, it's also a chance for students who study environmental sciences to watch and record real-time interesting phenomena in actual world. The program I will introduce today is Field Work in Environmental Studies 1, which is a required subject to first-year student for the School of Environmental Science in the USP. We have Field Work in Environmental Studies 1 and 2 for first-year and second-year students, respectively. They are common curriculums in all departments of our school. These programs are managed by teachers from 4 departments: and we interact these programs in teams. That means we treat these education programs not only from the viewpoint of the natural sciences, but also from social sciences. Through these programs, students are required how to observe environmental process or environmental problems from multiple viewpoints. The course of field works in Environmental Studies 1 consists of 4 themes. Each takes 3 to 4 weeks. All students must take all 4 programs and complete 15 weeks course in total. Here I listed 4 themes in usual year. Students work in and outside of our campus to find, record, and think about environmental problems, both from viewpoints of natural and social sciences. In usual years, our team organized the fourth theme, namely minor restoration of channelized rivers. These photos are the program done in 2019. In the first week, we take students a river which is artificially channelized and poorly improved, and they observed it. In the second week, the students make a plan to restore the damaged river. In the third week, they make an on-site trial of river restoration using some temporary materials such as sandbags. In the last week, they make a presentation about their plan and the result of on-site trial and write a final report. Again, this is a plan of our theme in usual years. However, we can't conduct this program in 2020 because of the pandemic, so we must make another online program instead of usual field activity. Next, I will explain the status in Japan in spring 2020. We were under a soft lockdown in which we can do only online classes in all schools, but people were not forced, but just recommended to stay home. Therefore, there were no problems to go out of home to uncrowded places. And we had room for feedback in one's resident area. This is the online feedback program in 2020. Can you find any changes of waterfront around your home? In this program, we assigned students a field work around their own home instead of the campus. We obtained a hint of this program from many news under a lockdown. As you know, many world news reported environmental changes under a lockdown. For example, in Venice, Italy, seawater turned to clear because the water turbulence by gondolas decreased in lockdown. In the US, birdsong increased because of the decrease of human noise. Such environmental changes following the decrease of human activity were reported from many countries under lockdown. Therefore, lockdowns can be regarded as a global social experiment, reducing the load of human activities on ecosystems. It's a quite unique chance for environmental scientists to observe how ecosystems react to this special event. So we expected that students can record and memorize the environmental condition under lockdown through careful observation of environment in their resident area. This is the motivation of our program in 2020. Here I write the schedule of 4 weeks feedback program. This is an announcement to a student in the introduction of the course. In first year, with a textbook and online slides to understand the aim and method of this program. A Zoom meeting room will open during the class time to test Zoom and to ask any questions to teachers. In second week, do feedback around your home and submit a short field report, and also prepare a short presentation within 4 slides for the next week. In third week, make a short presentation on Zoom in groups consisted of 6 to 7 students and receive advices from teachers. In fourth week, do second feedback based on a teacher's advice and write a final report. These are notes to students for field activity in a lockdown condition. If one doesn't want to go out of home, one can select the options of virtual feedback using online maps such as Google Maps. These are the examples of waterfront around us. Students can select any waterfront, not only lakes and rivers, but also rice fields and artificial waterfront and irrigation pond, etc. Next, we showed students the element of environment of waterfront. Here I list both natural and man-made elements and called their attention to the interaction between them, from which many environmental problems arise. These are examples of visual environmental problems in waterfront and an example of the solution. Under lockdown condition, many waterfronts was expected to less crowded than usual. However, in particular in urban areas, waterfront may be crowded than usual condition because of people who want to recreate in open space. Thus, environment can be changed both for the better and worse under soft lockdown condition. Students were required to give a question in their report on their observation, whichever they found a better change or a worse change. This is a part of one student's report. She observed an artificial creek out of a shopping mall and found there were very small number of cars and people than usual condition, and almost no trash during the lockdown. Her report clearly showed that trash on street directly correlates to number of persons who visited there. This is another report showing a lot of cigarette butts on the coast of Lake Beaver. These were thrown by anglers. In the lockdown, more anglers visited to Lake Beaver than usual for open space recreation. However, this movement caused another environmental and social problems. Another student reported that so many anglers' cars were parked on street around Lake Beaver. Because all public car parks were closed during lockdowns, they parked their cars on street illegally. Not a few cars were from cities. Not a few cars were come from cities far from Lake Beaver. For example, from Nagoya and even from Yokohama, suggesting long distance travel by some people, which was advised to stop during lockdown. These were post-class comments by students. There were a lot of positive comments by students saying it was a nice chance for them to go out of home and observe environmental problems by themselves. On the other hand, there were also a few negative comments against the real feedback under lockdown and insufficient communication with teachers in remote classes. But anyway, our program D in this table can get highest post-class evaluation score in almost all points in all 4 themes of the field works. Thank you for your kind attention for my presentation, and I am very happy if you can get any hints for the education plan in your remote classes. Thank you.
Thank you very much, Urabe Sensei, for sharing to us your experience in handling field work despite of the lockdown there in Japan. So let us now proceed to our last invited speaker for the day 1 of the UST-USP Symposium. So our next speaker is actually a young emerging freshwater biologist in the Philippines and a member of the Department of Biological Sciences in UST. He was able to finish his Masters of Biology at the University of Santo Tomas in 2019 with the topic of using benthic macroinvertebrate assemblages to assess anthropogenic impacts and protection status in the 2 watersheds of Laguna de Bay, Philippines, where he graduated with the highest distinction honors or summa cum laude. His research interest includes stream benthic macroinvertebrate ecology and taxonomy, freshwater food web structures using stable isotopes, and fish parasitology and aquatic entomology. At a very young age, he have been able to communicate his work and be able to publish them in reputable high-ranking journals at his field. Currently, he serves as a faculty member that handles the courses on STS, Environmental Science, Biostatistics, and Ecology Lab offered by the Department of Biological Sciences. Ladies and gentlemen, to present his talk entitled Erect Gears Towards Understanding and Managing Socio-Ecological Health of Watersheds in the Philippines, a DPSIR approach, let us give a warm round virtual applause to Mr. Alfredson M. Peralta.
Hi everyone. Thank you to the organizers of UST-USP Symposium for inviting us to present our experience in EREC project. EREC project have used gears to understand and suggest management towards socio-ecological health of watersheds in the Philippines, specifically in Laguna de Bay watershed and its sub-watershed, using a DPSIF framework. And together, or on behalf of the entire team, I'd like to share with you our outputs and how we were able to understand these watersheds. Okay, so, like what I've mentioned, we have 4 gears that we get to use as a core of the project, and it enables us to understand and eventually suggest management towards or attaining social ecological health of the watershed. So this presentation would mostly show you this part here where I'll be highlighting how we were able to understand nutrient cycling, how it enhances biodiversity and vice versa, and how it could affect the community activity at, for them as to protect it or how they can benefit from it, and eventually how can this empower them and be a factor to their well-being. So this is technically the framework of the ERIC project led by Dr. Noboru Okuda. Okay? And I'll be using a DPSIF framework so that we get to properly picture out how we were able to understand the watershed ecosystem. So from the human activities as the drivers and how this gets to um, give pressures, particularly environmental pollution, and what would be the status of the watershed ecosystem or the health, and how this can eventually impact, okay, and show environmental disturbance to the entire ecosystem, and how us can produce action plans, suggest action plan, so that we get to address the drivers to pressures in the state and even the impact that we were able to observe. So we have specified already and hypothesized that in terms of drivers, we anticipate, we recognize the contribution of land use and human population definitely, and how this can affect or intensify deforestation and nutrient loadings, and thereby allowing or affecting water quality, and showing or using by indicators we get to reflect this status or state of the watershed, and how this eventually impacts biodiversity or specific species or community of different community in the ecosystem. And lastly, how we can suggest policies for sustainable governance or at least management for this watershed ecosystems. So as you can see, we have investigated Laguna de Bay, and we then further looked at its sub-watersheds, Marikina watershed and Silang Santa Rosa sub-watershed. And for the onset of this presentation, I'd like to quickly show you the status of the catchment. And this was done at the onset of the project, where we've seen spatial patterns of water quality parameters across the entire Laguna de Bay. Okay. Well, if you're familiar with Laguna de Bay, it's the largest lake in the Philippines. And it being large, we have specified different bays inside the lake itself. So there's central, east, and southwest. So based on water quality parameters that we have measured, we get to see clustering of sites. Okay, and it reflects the status or the condition of each bay. And this is mostly attributed to the total nitrogen and total phosphorus loadings. And again, like what we have hypothesized, this is driven by different land use. For example, total phosphorus here is driven by built up areas or increasing fraction of built up areas, and total nitrogen is highly related to the increase of cropland areas. Okay, and aside from the water quality parameters, we have also monitored biodiversity, to be specific, mollusk and zooplankton diversity on the onset of the project, that is in 2015. And clearly, as you can see here, It is consistent that there is low biodiversity across the sites that we have sampled, both for mollusk and zooplankton diversity. And this is still, again, we were able to still observe this in 2017 based on mollusk and zooplankton distribution. Okay, but there is a consistent or weak patterns of community assemblages in 2015 and 2017. And this consistent low biodiversity are again, or is again attributed to various environmental factors that we have seen before in 2012 and in this data. To be specific, we try to look at the response of mollusk and the environmental parameters that we have sampled. And again, we have seen insignificant variation in terms of their diversity because it's in the entire lake shows consistent low biodiversity. And the same is true with zooplankton diversity, although we get to see some to be resilient to harsh conditions like increased total phosphorus, higher temperatures, and lower dissolved oxygen. We have seen how invasive Arctodioptomos dorsalis has the ability to survive such condition and be able to displace other zooplankton species. Okay. And aside from these patterns, we also use stable isotopes to assess and look at the food web in the lake using carbon and nitrogen stable isotopes. And how here we have used bentopelagic species like tilapia or Oreochromis niloticus to look at its food sources and how these primary producers here from phytoplankton and EOM or epiphytic organic matter or periphyton would be the source of nutrients of zooplankton, benthos, mollusk, and bentopelagic species respectively. Okay. But aside from this, we actually have observed more than the food web structure. Rather, we have used the nitrogen and carbon stable isotope signatures to look at if we will get to observe patterns or clustering of sites according to the condition of different days in Laguna de Bai. And that is true. We were able to see similar patterns or clustering based on the water quality parameters. So this is a reflective of the condition of the lake, not just in terms of water quality parameters, but at the carbon and nitrogen stable isotope signatures. And lastly, we have also observed how, interestingly, actually, how human population density, increased HPD, would tend to increase the nitrogen stable isotope signatures of the following organisms that we have sampled. As you can see here, increasing HPD would show increasing nitrogen, stable isotope nitrogen signature as well from the primary producers, consumers, and to the bentopelagic species like tilapia. Okay, so all of this observation were significantly correlated, as you can see on screen. So with this status of the lake, we tried now to ask ourselves what are the major contributors of such condition in the lake. So again, the most practical ways to look at to the tributaries, to the different subwatersheds draining to the lake. So first, we wanted to investigate how nutrient pollution and deforestation would affect this condition in the lake and in the river itself. And aside from this disturbance, we want to also highlight how we can further protect and manage our river ecosystem, as well as how it can eventually affect the entire watershed. Okay. So let me now show you our study ecosystem. So first, this is the Silang Central Subwatershed or SSRS. So I hope you get to remember how the sites are oriented here, because I'll be referring this whenever I show the results. Okay. So this is the upstream sites in SSRS. This is the midstream and the downstream as it drains to Laguna de Bai. Okay. While for the other subwatershed, which is Marikina watershed, okay, interestingly, although this has the same urban developments, the Marikina watershed, the upper portion of it has a freshwater protected area, which has recently established around 2011. Okay, so this is the Upper Marikina River Basin Protected Landscape, or the UMRBPL. Okay, so from here we try to investigate and sample for anthropogenic and environmental variables. Okay, aside from water quality parameters, we try to look at also canopy openness for each site that we visited. And of course, we sampled bioindicators such as macroinvertebrates. And again, these are very crucial and important for us so that we get to see how these organisms are being affected by various environmental conditions. And then we analyze our data using univariate and multivariate analysis or modeling. So similarly with what we did from Laguna de Bai, we tried to understand first how the nutrient cycling happens in each study ecosystem that we have. And you can clearly see here how nitrates and total phosphorus gets to shift from the upstream to the downstream, how the patterns gets to shift from high nitrates up to the down to the lowest values of nitrogen concentration and nitrate concentration, and vice versa for total phosphorus. Okay, and we have a use oxygen isotope ratio of phosphate to further understand the possible sources of this phosphorus or total phosphorus. And we have clearly see the how this phosphorus sources attributed to different land use, specific land use types like industrial, residential land use. Okay. Aside from looking at the phosphorus sources, we also tried to look at the nitrates, or to be specific, where we can attribute those nitrogen pollution in the river. So here we have monitored from 2016 to 2018, and consistently we have observed that this is attributed to manure and septic waste. This provides us an information how direct effluents from the households get to really affect the environmental condition of the streams. Okay, so using GIS, we try to look at the best descriptor in terms of increased total phosphorus across the sites that we have sampled, and we have seen how human population density, or HPD, increase of this variable would tend to increase total phosphorus as well. And this is highly related not just with what we have observed, like what I have explained before, but this is also attributed to phosphate-based detergents that we use. And this speaks of the poor wastewater treatment facilities that the subwatershed has. Okay. And also we try to look at how total phosphorus would then affect other critical environmental factors in the water ecosystem, in the river or streams ecosystems, and that is the dissolved oxygen, for example. And increasing PP would tend to deplete the dissolved oxygen in the environment, and as you know, this is very critical to the various organisms in the stream. To be specific, I'll show you later what are the bioindicators that we have used for this. And this is attributed to the increased bacterial respiration activity, okay, in the environment, since total phosphorus tend to enhance this bacterial activity, or bacterial respiration activity rather. Okay, so for that we try to look at bioindicators and if it will reflect the condition of the stream ecosystems, and that is true. As you can see here, you get to observe colorful charts then versus less colorful charts as you go downstream. And again, this is attributed, okay, to the water parameters that we have investigated, specifically those aquatic organisms that are aquatic insects that are sensitive to dissolved oxygen and canopy openness. So this is these organisms are favored with high dissolved oxygen, with low canopy openness, and less ammonia and urban loadings. So here We can see here the sensitive EPT taxa or the aquatic insects that we were able to sample, and they indicate good water quality in those sites, those upstream sites in the river ecosystem. And on the other side of the plot, you get to observe those organisms, or at least few organisms that would be resilient to, or would be very tolerant to harsh condition like lower or depleted dissolved oxygen or anoxic condition and higher canopy openness, okay, and higher nutrient levels, okay, like chironomus or bloodworms that you can see here on screen. So aside from bioindicators, we try to look at the whole picture and by looking at their biodiversity, and we have observed how dissolved oxygen is critical to maintaining high biodiversity, as you can see here in figure A. While for the other side, this is also true for the richness that we get to observe in the ecosystem, species richness or S. Okay. Aside from that, okay, we want to highlight how important the riparian vegetation in the stream ecosystem. We are using canopy openness as a proxy for that. So as you can see here, increasing canopy openness would tend to decrease the EPT richness and EPT density. If you can still remember the aquatic insects I have presented to you, those stand for Ephemeroptera, Plecoptera, Trichoptera organisms. You don't need to remember them, but you just want to understand that these aquatic insects depend to the allochthonous food sources that they get from the canopy or from the riparian vegetation in the streams. Okay, so we tried to do variation partitioning to determine which really or which highly affects the entire ecosystem, and we get to see how nutrient and organic pollution would really affect the entire, or would significantly affect the entire Silang Santa Rosa subwatershed. Well, aside from being able to highlight nutrient pollution, deforestation, we also want to highlight how we get to further manage it, and by using freshwater protected area, okay, and what is the benefit of such ecosystem. So allow me to show you the picture. This is just a small portion of the entire freshwater protected areas, the FPA. I'm happy to show you this picture right here was actually used by Pacific Science as a cover of their issue. Okay, and clearly you can see how Richard riparian vegetation from the protected or freshwater protected area would tend to, of course, have better water quality or better steam environments versus those unprotected sites, okay, where you expect higher urban material loadings from domestic and agricultural land use. Okay, so here is the data that we have gathered from this site for the entire freshwater protected area rather. And we try to analyze that further by using ordination analysis, and we clearly see how protected areas in terms of their water quality would differ from unprotected areas. Okay? And the same is true when we try to look at the benthic macroinvertebrates as bioindicators. We get to see higher diversity in protected areas than unprotected sites. And these are just the samples that we were one of few of the samples that we were able to obtain from this FPA or UMRBPL. Okay, and based on their diversity, we get to observe again higher rates of diversity in such ecosystem. And this is still for us to see or differentiate the various biodiversity indices and EPT and EPT indices, okay, differentiating protected and unprotected areas. Okay, so from this, we just have few takeaways from maybe few or more, but allow me to just give you the conclusion of the project, where we see the first empirical evidence to show how human population density can be a predictor of urban stream syndrome effects, or what we have seen how urban loadings would tend to affect the entire stream ecosystem. And biodiversity loss in urban tropical stream systems are highly driven by phosphorus eutrophication, dissolved oxygen depletion, and increase in canopy openness. And we also want to highlight the effectivity of FPAs in environmental maintenance and biodiversity conservation in urban tropical stream systems amidst rapid economic and population growth, like what we have here in the Philippines. And as you can see, this is the entire picture of Laguna de Bai, okay, with the major contributors of from various tributaries or from various subwatersheds. Okay. With that, I conclude my presentation. If you have any questions, feel free to approach me. Thank you so much for listening. These are the recommendations of the project. Thank you.
Thank you very much, Mr. Peralta, for that very interesting talk. And the information that you've shared is really amazing, for especially for me who doesn't have any knowledge about aquatic ecosystem. I'm enticed now to study more about our lake ecosystem. All right. So thank you very much for the talk. So now, once again, I am opening the online floor for our guests, professors who are here right now to ask our distinguished and esteemed speakers about their presentation about the aquatic research and ecology. So you can just type your questions on the chat box, or if you want to do it live, I do really encourage our students who are here, our colleagues. Yes, Sir Masahiro Maruo.
Thank you for the introduction speaking. I have to ask about the phosphorus to every lesson.
はい。
Oh,
丸部先生。
yes, お久しぶりです。So I want to know the original compounds included in detergents because so in Shiga Prefecture, for about 35 years ago, there's a news about the phosphorus contaminated detergent, in especially in those area. In that case, triphosphate is used in the water softener in the detergent, and then it causes the eutrophication in lake in southern part. Now, what kind of compounds used in the detergent?
Yes. Thank you for that question, Maruo Sensei. Yes, you are correct. To confirm, there is still phosphorus compounds or the detergents that we use at home are all phosphate-based detergents. That's why we highly attribute it to what we have observed, increased total phosphorus, especially that we have observed direct effluence from the households to the river. So it's very likely that this is from those phosphate. But to be specific of what phosphate or what specific compound of phosphate included with the detergents, that's something that we did not look at. But that's something that we tried to use also by using phosphate isotope, oxygen isotope ratio with Dr. Ramirez's team. Yes, that's why we are also highlighted the legacy in Japan, where you guys, or especially in the northern region, as you've mentioned, where you were able to really remove or do away with the inclusion of phosphate or phosphorus in the detergent just to address this concern.
Thank you. So, in case Shiga Prefecture over Japan, now we don't use this type of phosphorus compounds in the detergents. Now they are shifted into the other compounds. For example, some solids that has an ion exchange capacity from calcium manganese into sodium. But I think it depends on the cost for production of detergent.
That's true. That's true. That's something that we also look at, that how we can try to reach to the manufacturers, to the big companies behind these detergents that we have here in the Philippines. I hope this is a good first step or baseline information where we can base future management.
Mm. Thank you very much.
Thank you, Maruo Sensei.
Thank you, Maruo Sensei. Do we still have other questions in the house? Ferdino.
Yeah,
Yes.
before I ask Mike, I have a question for Urabe Sensei. But before I ask, I just want to share because JC and I stayed in the same house in Ishidaracho. And I remember that the detergent that we use for laundry and the shampoos and the soap that we use for taking a bath are specialized because the water that we use for this laundry and for taking a bath goes directly to the lake. So I think these are specialized chemicals that are used. So they do not harm the lake ecosystem when they enter to the waterways or into the body of water in Lake Biwa. So yeah, because we live right beside Lake Biwa, that is really just a few meters away from Lake Biwa. So that's something that I learned when I was staying there. My question for Urabe Sensei is, you mentioned, Sensei, that the students go out of their houses or their within their areas, and then they do their field observations and where they stay. I would like to ask, do they do this as individuals or do they work as a group? Because I think the situation for in our laboratory classes, because the students always work in groups. So I would like to know your how you are able to accomplish this. I would like to know how you are able to do this individually, or do they work? Do they do this individually, or do they work in groups?
Yeah, they worked individually because their houses separated. They usually live different towns and cities. So they had a chance to made a presentation and discuss each other in groups in Zoom. We set around about 15 minutes discussion time for each group. And we also advised how to observe and record around during their field works into class.
I just want to know, Sensei, how many students are there in your class?
50
Good.
in total, very, very large. And in total, 5 teachers manage each class. So we set 2 Zoom rooms for discussion, and each student and we divided whole students in 8 groups, 8 small groups, and so we stayed the Zoom room for several hours, but students come and go out for each groups.
Okay, that's very interesting, Sensei. Maybe we can adapt a similar approach for our laboratory classes. Well, hopefully we will go back to we'll start going back to the original setup. But since we're still not there, so maybe we can adapt something similar. Thank you, Sensei.
Thank you, Sir Dido, for that question. All right. So do we still have other questions that you would like to ask?
いいですか?
Yes, since Rabbi Sensei.
Yes, I have one question too easy. Yes, you measured canopy openness as one of the factors affecting the なんだっけinsect community.
That's right.
But,
Yes,
yes,
that's right.
so,
Urabe
Since
Sensei, you're on mute. You're on mute. Yeah. Okay.
sorry. えっと、do you think what sorry ごめんね。what environmental effect what environmental factor the canopy openness is affect? For example, なんだっけ、land use or water temperature itself.
Thank you for that. Thank you for that question, Orabe Sensei. Yes, we tried to use those different factors. Actually, aside from canopy openness, we tried to look at the effects of various nutrient loadings, temperature, even pH. And when we tried to do multivariate analysis, the best descriptor for the specific EPT group, or Ephemeroptera, Plecoptera, and Trichoptera, Tubicera. So those organisms are best described, or the variations of those organisms are best described by canopy openness. And then when we try to look at it, definitely most of those organisms that we have collected from EPT are dependent on allochthonous food sources. So those coming from leaf litter of the canopy openness, of the canopy rather. So the richer the riparian vegetation, the better allochthonous food sources we expect them to receive, especially those scrapers, collectors, gatherers belonging from those functional feeding groups from FFGs, from EPT. So, but definitely the environmental factors like land use, using GIS, the nutrient pollution, were also correlated to these organisms. But we just chose among those parameters what would be the best descriptor for the specific group of aquatic insects of EPT, and that is canopy openness.
So it's a parameter of litters coming into the river ecosystem.
Yes, that's right.
Okay. Thank you so much.
Thank you.
All right. I thank you, Urabe Sensei, for that questions. All right. Okay. Can we still accommodate one more question? All right. Well, okay. Just maybe for it will also be my wrap up, especially for our Japanese colleagues here. I'm very interested in the time of pandemic. How were you able to handle your research groups, especially your advising and your mentoring on your graduate students, especially now that we are also going to handle some research for our bachelor's students in our university, in our department. Maybe you can share to us some of your advice on how you were effectively able to manage your groups, especially those that needs, for example, field work to get or acquire their data. Anyone from our Japanese colleagues here, or maybe some of our seasoned faculty members who also like to share their experience during the pandemic?
Okay, so may I start?
Yes.
The, yeah, in my case, I have one master course student and 5 undergraduate students in my laboratory. And the one master course student came to the laboratory once a week. At that time, he concentrated for the isolation of epiphytic bacteria from invasive aquatic weed Egeria densa. It takes time. First, he streak the microorganisms. After 1 week, he checks the the evaluation of the plate. It takes time. And other times, he concentrated the reviewing the literatures. They also, undergraduate students, they almost concentrated in the literature works. After, in the end of July, they are permitted to come to the laboratory. And after the detailed health check, they could started working in the laboratory, in my case. Any other instructors in my university?
Yeah. Or maybe, for example, to our Indonesian colleagues who probably also were able to do some research advising. Maybe you can share some of your tips on how were you able to conduct your research mentoring in this new normal setup. Mhm.
うん、なんだったかな。うん。
Oh, very hard question, no?
Yes, yes. I think there is a little bit difference between undergraduate student and master course or doctor course student. And Urabe Sensei is taking care of so many master course student and doctor course student, especially some of them are from foreign countries.
Mhm. Yeah.
Urabe Sensei.
Okay, but I guess it's really still a challenge for many of us who to mentor and to advise students, especially now
We
at this
ask the
pandemic.
graduate students.
That's also a good
I see
thing.
some
Maybe we
graduate
can
students
interact with
here. I see
some.
Arvin here, which is under Urabe Sensei's laboratory.
So maybe we can also get some feedbacks from the students how were they were able to be mentored at the time of a pandemic, because it will be very helpful for us to also use this platform to share our experiences, and if there are things that we need to improve, especially now that we're still on the middle of this pandemic crisis.
I would just want to uh, ex, share my experience as a research student and during pandemic. So it is a really difficult situation. So, but for me, I just usually do my paperwork or my research work at home. But whenever I do laboratory work, so I just go to school. So during that time, so mostly I just do paperworks now because I'm my writing my manuscript and publication. So, but within this coming school year, so I will be performing another experiment. So I will have to go to school probably uh, two times or three times a week. So yes, thank you very much.
Right, good luck to your publication, Arvin, and
Thank
we're
you,
looking
Arvin.
forward for that papers to be published soon. All right. Okay. So are there other feedbacks or
I think
commentary?
it will
Yes, Steve.
depend on the type of research.
Mhm.
If you're doing research that won't really require, or it depends either on the type of research or maybe on what stage of the research process you are in. In the case of Arvin, he's already in the writing stage, so maybe he doesn't need to spend that much time in the laboratory. Or maybe the type of research that you are doing would require you only to spend a day or two in the laboratory, more out in the field. But for example, if I did my research requires me to stay in the laboratory every day. So
Right.
to observe my specimen. So I think it would be very different. I'm lucky that I have no active experiments during this time, but I can only imagine the situation of other students. For those who study microbiology, of course, they need to check on their specimens every now and then, every 24 hours. So I think it depends on at what stage of the research process you are in and what type of research you are doing.
Mhm. But
Uh,
one thing is for sure. Yes,
Yeah,
Arvin.
sorry, I just want to add. So last year during so the start of the pandemic, so our lab adjusted, so we set a certain time schedule per student. So we have to book the lab if we are going to use it, or so it really depends on the size of the laboratory or the members. So we have to set schedules and prepare, and yeah, thank you very much.
Mhm. Well, one thing is for sure, we're all wishing that this pandemic soon to end, so that we are already excited to go back on our face-to-face classes, and at the same time to be more active in our research productivity soon, hopefully. All right. With that being said, that summarizes the afternoon session of our fourth UST-USP Symposium. And I'm very happy that the afternoon session went very smoothly and the active participation of most of the people who are currently here. So we thank you, and we do appreciate and hopefully tomorrow it will be much more interactive, since tomorrow we have a lot of activities that is in store for everybody. All right. So at this juncture, I will now give the floor back to our organizing chairperson of the UST-USP Symposium, Dr. Jonathan Carlo Briones, for some announcements.
Thank you very much, Dr. Nikki, for the great moderating activity. So we would like to take this time to thank everyone who came into this day one session. So as we've noted, this is a collaboration, a joint symposium by the University of Santo Tomas and the University of Shiga Prefecture. Each of us having our respective organizing committee. So from the side of the University of Santo Tomas, we would like to thank everyone who came, all who are the invited speakers, our students, our guests, and most especially our co-collaborators in this event, all of the teachers from the University of Shiga Prefecture, Harada Sensei, Maruo Sensei, Takahashi Sensei, and most especially our co-chair for this joint symposium, Professor Misako Orada. So, and we thank all of you that even though in this time of pandemic, these various challenges that we have, you've opened up this collaboration first with our institutions, but now together with other institutions like with Bogor University in Indonesia, and our representatives as well in Vietnam, and some of our representatives from the Philippines, for example, in Iloilo University, who have also who will also present tomorrow. So this just shows that our collaboration is ever fruitfully developing more. And we hope to see you all once again, once all of this is, once it's safe for everyone. So hopefully this activity may deepen our friendships and relationships, our collaborations, and we bid you a very good day. And with that, we would like to say thank you very much and see you tomorrow. So for tomorrow, please take note. We will have our activity starting at 8 o'clock Philippine time. So if you can, please come in maybe 15 minutes or 10 minutes just before that. And we promise that we will be here in our Zoom. So to end, may we ask everyone to turn on their cameras and even their microphones. Okay. So you can also turn on your microphones. So with this one, you would like to thank everyone, and let's give everyone who participated a warm round of applause. Thank you very much for the success of this first day of the 4th UST USP joint symposium. And see you all again tomorrow. Thank you very much.
Thank you
Thank you.
so much.
Thank you so much.
Thank you po.
Thank you.
See you tomorrow.
See you tomorrow.
Thank you.
Bye.
And share your screen para you can see the others more. Nag-aalis na pala.
Okay na ba yung recording?